Good morning, children. So in the last class we were discussed about the human genome project. In that one we were discussed about the aims, methodology and even procedure as well as the salient features of the human genome project, isn't it? So in this class we have to discuss about the another important topic, so as well as is the last topic of this chapter. So that is nothing but DNA fingerprinting technology. Okay. See, you must have heard about this terminology, the number of cases. Normally, even the science students, if they don't know about this one, they will, generally they will think that only collection of the fingerprintings. But, so this is not like that. Okay, so that is also one of the collection of the samplings. But, so here the question arises, then what is the meaning of the DNA fingerprinting exactly? Okay, so let us discuss about the DNA fingerprinting technology, what are the steps and applications. Okay, so how it is utilizing in the society, all those things one by one. So let us start the discussion. The DNA fingerprinting technology, it is also known as DNA profiling. Okay, yeah. As I said, first of all, the question arises, how can you define or what is a DNA fingerprinting technology? The DNA fingerprinting is comparison of the DNA from different sources to establish the identity. Okay, so that means it is a technique to identify the similarities and differences of the DNA fragments of two individuals. Okay, so what they will do during this technique, it is an analytical technique used in detecting and comparing specific nucleotide sequences in DNA of individuals and thereby identifying the similarities and differences in such sequences. Okay, are you getting? And here what is the process actually it is involved, the principle behind the DNA fingerprinting technology. The generally while we are studying about the basis, I told you DNA basis, there will be containing the non-coding DNA. Okay, the non-coding DNA, it is also known as junk DNA of an individual, consists of repeated nucleotide sequences. So they are also called variable number tandem repeats. In the short form, it is also known as Vntr, and which are different between two individuals is used in identifying different human beings. So that's why each and every person in this world, they are unique. Okay, they are unique. They are all different from one to another, except the homozygous twins. And the method employed in DNA fingerprint is distinction fragment length polymorphism. So this one they will also ask for 1 mark question. What is the method employed in the DNA fingerprinting technology? The method employed in DNA fingerprinting is RFLP, that is nothing but the restriction fragment length polymorphism. Okay, and this DNA fingerprinting technology was developed by Alex Jeffrey from Leicester University, okay, UK. So in 1985, because of his the discovery of the DNA fingerprinting technology, even he is awarded Nobel Prize. Okay, that was the all-time the wonderful discovery in the field of science. Okay, so here whenever you see the DNA fingerprinting, so how it will be the basis. See, as I said, see, generally the DNA carries some non-coding repetitive sequences. Each and every portion, okay, in each and every portion, whatever they contain in the genetic material, the DNA carries some non-coding repetitive sequences, and the number of repeats is specific from person to person. Okay, so that is it is not almost only a few non-coding repetitive sequences will be present, but so number of the repeats is specific from one person to another person. Here only you can identify. So there are the the 3 repeats they have given. So the person 1, person 2, person 3. Okay, if you see from the beginning from the whatever the sequences, GA, TA, GA, TA, GA, TA, GA, TA, like that up to 8 repeats are similar. Okay, so in the next cases, in the second portion, in case of the second portion, it's about 9 repeats are similar. Okay, and what about the next? In the third case, the third portion also, the 10 repeats are similar. Okay, so that means the number of repeats is specific from person to person, but still DNA carries some non-coding repetitive sequences. Okay, and the very important one, whatever the repetitive DNA can be separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the small peaks called satellite DNA. So this is very important. The repetitive, whatever the DNA, the repetitive DNA can be separated from bulk genomic DNA as different peaks during density. Okay, and so what basis they will separate? Based on the density gradient centrifugation. And the bulk DNA from a major peak and the small peaks are called satellite DNA. Okay, so in this, the centrifugation tube, you can identify how it will be. The bands will be diffuse with respect to the major peak. Okay, isn't it? Are you able to follow? Yeah. So this is the the density gradient centrifugation, during which we will separate the bulk DNA from the major peaks and from the small peaks. And after one, the satellites, as I said, the satellite DNA is classified as microsatellites and still mini satellites, etc. like that, based on base composition. Example, whatever the adenine and thymine are rich, or based on guanine and cytosine rich. So like that, the length of the segments and the number of repetitive units, so they will be separated. And a DNA sequence which is tandemly repeated in many copy, a number is called variable number tandem repeats. So here only I hope you can identify how it will be. So that microsatellite short tandem repeats and mini satellite Vntr, that is variable number tandem repeats. Okay, and the Vntr belongs to mini satellites in the DNA commonly. So here only you can identify how it will be. The microsatellite, the short tandem repeats, and mini satellites, that is variable number tandem repeats. Like that, we can differentiate as well as identify. Yeah. So and in a portion, any portion, in a particular portion, the copy a number carries in each chromosome. 2 alleles, that is from the paternal and the maternal. You know what is the meaning of allele, right? Yeah, I hope you remember. Yeah, the 2 alleles, the parental and maternal, that is a paternal and a maternal of chromosome, also contain different copy number of Vntr. So that here we can easily identify the copy number of Vntr from the paternal and maternal 2 alleles of a chromosomes. Are you able to follow? Yeah, the microsatellite as well as the mini satellites with respect to Vntr. And finally, the Vntr is specific from person to person. As I said in the beginning only, it is different. So even though we are all humans, even though between the men and between the humans, how can we differentiate? So based on the Vntr. Okay, the Vntr is specific from a person to person. Okay, and the size of the Vntr varies from 0.1 to 20 kb. That is nothing but kilobase pairs. Okay, or kilobases. Okay, so that will be varies from one person to another person. It is not similar in all the cases. It is different. The size of the Vntr varies from one person to another person. So between 0.1 to 20 kilobases, so that we can identify with respect to different peoples. Okay, yeah. And any differences in the nucleotide sequence, inheritable mutation. I told you during the sexual reproduction the mutation is very important, that is called the positive mutation, isn't it? During sexual reproduction we can identify that is nothing but variation. So that means any difference in the nucleotide sequence, inheritable mutation observed in a population is called DNA polymorphism. This is very important. 1 mark questions also they will ask, what is the meaning of DNA polymorphism? Any difference in the nucleotide sequence, inheritable mutation observed in a population is called DNA polymorphism. The variations at genetic level, okay, is also known as DNA polymorphism. Okay, here only you can identify, they have given the right side in the slide. And remember, the polymorphism have great role in the evolution and speciation. What is evolution? You know very well that a small, the gradual changes took place in millions of years. And what about the speciation? A single step mutation leads to large variation, is called speciation. Okay, and see, I was telling about the polymorphism, isn't it? So this polymorphism is higher in non-coding DNA sequences because mutations in these sequences may not affect an individual reproductive ability. So this is very important. Even though the mutation happens in the each and every level of the during formation of the filial generation, so it will not affect, or it should not affect, the reproductive ability. How? Because the polymorphism is higher in non-coding DNA sequence, because mutation in these sequences may not affect an individual reproductive ability. So these mutations accumulate the generation to generation, causing polymorphism. So that's why, so we are all look different, we are all different from one to another, of course. So if you see, there are different type of the the males or the females in the society, how they are all different from one to another. So this is because of the polymorphism. Okay, the poly means many, morphism means the morphological structure. Okay, yeah. See, after this one, the question arises, how we will perform this, the process, or what are the steps involved in DNA fingerprinting technology. Okay, so the DNA fingerprinting technology in generally we also known as southern blotting techniques, because nowadays whatever we are using this technique, okay, so that is a popular one, the southern blotting technique. Okay, you can also ask me, sir, what is the southern blotting technique? See, generally during the the DNA fingerprinting or the profiling, the single standard DNA fragments present on the gel plate are extracted on the nylon mesh or nitrocellulose membrane by the process. So that's why it is also known as southern blotting technique. Okay, so let us discuss what are the steps involved in DNA fingerprinting technology. And that one, the first step is nothing but isolation of the DNA. Isolation of the DNA, it is also known as collection of the samples. Okay, isolation of the DNA or collection of the samples. What it will be involves. See, generally whenever the process begins, what we will do? Consider something, any just like a criminal offenses happens. Whenever the forensic, the team enters, what they will do? Okay, so that means they will do first collection of the samples. A sample of the blood, it may be semen. Okay, during rape cases generally they will collect the semen or the sperms also, and even the saliva, that is nothing but the spitting, and even the hair follicles. Okay, hair follicles, a skin. Okay, the skin may be present between the the nails. Okay, or any small pieces enough. Okay, and the bones, sometimes they may be burnt. Okay, even still they can collect the bones or any another tissues even in the dried condition. Okay, dried condition is collected. So this is called the collection of the sample. So that is nothing but the first step, isolation of the DNA. Okay, remember a living cell is enough. A single living cell is enough. Okay, so that they can multiply. In the further stages. Okay, so this is the first step. The next step is, okay, that is nothing but the digestion of DNA. So this is also known as the extraction of DNA. What they will do in this, the step, see, whatever they collected the sample, okay, so whatever the collected sample, either it may be, I told no, the blood, okay, a semen, a saliva, hair follicle, skin, bone, or any other tissues. So this sample is centrifuged. This sample is centrifuged at 2000 rotations per minute. Okay, 2000 rotations per minute to spin off and separate the cells, so that the cells are treated with enzymes to digest the plasma membrane and nuclear membrane to split open the nucleus and freeing the spaces. There is nothing but freeing the spaces of DNA. The sample of DNA is treated with REN. Okay, the sample of DNA is treated with REN. REN is nothing but the restriction nuclease enzymes to obtain the fragments of DNA of different lengths. Okay, so this is also called the digestion of DNA by restriction endonucleases. Are you getting? Yeah. And the next step is very important one, the separation of DNA fragments. Okay, the separation of the DNA fragments. So what they will do during this step? The DNA fragments are allowed. See, after the extraction of DNA, the DNA fragments are allowed to separate on agarose gel electrophoresis. Okay. The DNA fragments are separated on the gel plate according to their density gradient, where heavier DNA strands are settling at the bottom of the gel and the gel plate and higher fragments towards the top. So these DNA fragments are treated with alkaline solution to dissolve the hydrogen bonds which is present between the base pairs. Okay, so that to produce a single stranded DNA. So after the dissolving of hydrogen bonds, the double stranded will get converted into single stranded DNA. Okay, so this is very important. So if there will be no break or the dissolve of the hydrogen bonds, we cannot compare the fragments. Are you getting? Yeah, so that's what the separation of the DNA fragments is also known as by gel electrophoresis. Then treat the DNA fragments with alkali solution. Though what we use is a solution as alkali, sodium hydroxide. Sodium hydroxide to denature the DNA bonds in the gel into single stranded, so that the double stranded is get converted into single stranded. Okay, so these are very important. The next step is very important one. So that is nothing but the transferring of DNA fragment. It is also known as blotting of DNA fragments. Okay, the blotting of DNA fragments. See, this process in generally, as I said, in the beginning only. So this is also known as the extraction of the DNA fragments. So the extraction of the DNA fragments is done by using southern blotting technique. Okay, so they will also ask one more question. How we will do extraction of the DNA fragments, or what is the southern blotting technique? The answer is same. The single stranded DNA fragments present on the gel plate are extracted onto the nylon mesh or nitrocellulose membrane by the process called southern blotting technique. Okay, so how can you define the scissor blotting technique? The single stranded DNA fragments present on the gel plate are extracted onto the nylon mesh or nitrocellulose membrane by the process called southern blotting technique. Okay, so this is very important so that we can get the the extraction of the DNA fragments. Okay, so this is the fourth step. So the next step is very important one. So the tagging radioactive DNA probes. It is also known as hybridization using radioactive labeled Vntr probes. Okay, the tagging radioactive DNA probes or hybridization using radioactive labeled Vntr probes. What is the meaning of this? Okay, let me explain. See, after the extraction of the DNA fragments by the process southern blotting technique, the nylon mesh containing a single stranded DNA fragments are incubated. Okay, the nylon mesh containing a single stranded DNA fragments are incubated with radioactive probes. The probe is a radioactive synthetic DNA of no nucleotide sequences generated by a clone restriction fragments, so that the DNA probe binds to the restriction fragments on the nylon mesh. Are you getting what I am telling? See, this is very easy, so try to understand. So after the extraction of the DNA fragments by the southern blotting technique, we have to do the hybridization using radioactive label Vntr probe. It is also known as tagging radioactive DNA probes. What we will do during this one? The nylon mesh containing a single stranded DNA fragments are incubated with radioactive probe. So then the probe is a radioactive synthetic DNA of no nucleotide sequences generated by a clone restriction fragments. So the DNA probe binds to the restriction fragments on the nylon mesh. So this is the very important ones. And here the question arises, why we also call the hybridization DNA? See, as I said, the place the nitrocellulose filter paper in a southern of radioactive labeled single stranded DNA probe. Then the DNA probe binds with the complementary sequences of the DNA fragments on the membrane to form hybridized. So here only I hope you can identify the technique which is given in the slide, the radioactive Vntr probes. So then how it will be get converted into Vntr probes hybridized to DNA fragments. Are you getting this? The major one you should remember, the what we use here, the nitrocellulose filter paper is used to do. Okay, yeah. So this is what we call the tagging radioactive DNA probes. Fine. So this is the fifth step. And the last step is nothing but autoradiography. So autoradiography, it is also known as the detection of hybridized DNA by autoradiography. Okay. So what is this actually? What we will do? See, during this step, the last step, it is also known as autoradiography technique. An X-ray film is placed on the nylon mesh. An X-ray film is placed on the nylon mesh to obtain an autoradiography print. So that the X-ray film is then processed to obtain visible patterns of the bands of DNA. I hope you can identify here. An X-ray film is placed on the nylon mesh to obtain an autoradiography print. So that the X-ray film is then processed to obtain visible pattern of the bands of DNA. Okay. And now it is nothing but DNA fingerprint. Did you got it? Why we generally call this name the DNA fingerprint? A comparison of such two different DNA prints can provide information about the degree of similarities and differences. Okay, so that generally we use this technique. Nowadays we are using this technique because of the wide range of the applications in the field of the science and technology. Okay, fine. Yeah. So, and very important one, remember the DNA fingerprints differs in everyone except in monozygotic identical twins. I told you in the beginning also the DNA fingerprint is differ with respect to each and every person. So you must have heard about generally in films and all they will show. There are 7 peoples in the world, they may be look similar or exact copies of each other, like that you must have heard about the stories. But when it comes to the science, so those all cannot be believable. Why? Because I told now, based on the DNA fingerprinting technology or whatever the DNA profiling, till now it is concluded that the DNA fingerprint is differs in everyone except in monozygotic or identical twins. Only in monozygotic twins we can identify maximum similarities. Except that cases, almost all the peoples are different from one to another. Okay, then the sensitivity of the technique can be increased by use of polymerase chain reaction, that is nothing but PCR. So therefore the DNA from a single cell is enough for DNA fingerprinting. That's what I said. See, nowadays there are number of people, they will think that while they are doing any crimes, okay, so they will think that they must have not left anything, okay, proofs. But still I told no, a single living cell is quite enough. Okay, so that because of that single cell only we can identify, we can multiply, we can separate and extract all the fragments, so that we will get the complete information about that the particular person. Okay, yeah. So this is actually the process will be takes place in the form of the cycle. Okay, that is a denaturation, annealing and extinctions. So about this PCR process and all, while we are studying about the biotechnology, I will explain once again, because here only it is related to the DNA fingerprinting or the DNA profiling is given. Okay, yeah. See, this is at a glance what is the total exactly the process, the DNA fingerprinting technology. So it is a 6-step process actually. The steps involved in DNA fingerprinting, so what are those ones? The first what we will do, so that is a collection of the sample. It is also known as isolation of the DNA. The next step, the digestion of DNA by restriction endonucleases. It is also known as extraction of the DNA. And the third step, separation of the DNA fragments by gel electrophoresis. It is also known as separation of the DNA fragments. Okay, and the fourth one, transferring DNA fragments to synthetic membrane. So that is nothing but extraction of the DNA fragments. And the fifth one, hybridization using radioactive labeled Vntr probes. That is nothing but the tagging radioactive DNA probes. And finally the last one, autoradiography, the detection of hybridized DNA by autoradiography. So this is at a glance, so that you can remember. So this is one of the important question. If they will ask in the exam also, you should write all the 6 steps. Okay, so then only you will get the complete marks. See, and finally I told no, so why we are studying and why we are using all these techniques, because of its applications, because of its applications of the DNA fingerprinting. Nowadays we are using this technique. Okay, so let me to explain what are those applications of the DNA fingerprinting technology. The DNA fingerprinting technique is used in the identification of criminals like murderers, robbers, rapists, etc. Okay, so in general we can also say it is actually used as a forensic tool to solve the paternity, even the rape and murder cases also. See, you must have heard about there are a number of cases the paternal dispute arises. So there are a number of cases, in the 2004 actually, when the tsunamis happened, a child is displaced. Okay, that means due to the whatever the water current, the child is displaced actually. So there are two mothers actually, they were crying and they were telling that child is mine. Okay, then in such a cases, emotionally we cannot give the justice. So then those case, that case went to the court. There they conducted the DNA test, and finally they identified who is the real mother of that child. Okay, so that is nothing but it is employed in solving paternal disputes also. Okay, yeah, and a very important one. So it is useful. I told no. So in identifying the reunion of the lost children with their parents during natural disasters like earthquakes, floods, tsunami, etc. Okay, and it is useful in identifying the victims of plane crash, building collapses, fire breakouts, etc. So this is very important one. And another one is the diagnosis of genetic diseases, so that in the beginning stages only we can identify, and even to determine the polygenetic status of animals. Okay, polygenetic status of the animals. Are you getting? And the very important one, to determine the population and genetic diversities. So it is used in the population diversity determinations, so that we can identify what is exactly how the diversities happen. And okay, so here the one of the example they have given how to compare the solving paternity using DNA fingerprinting. So person 1 is likely to be the father of the child. Okay, so they will compare. Then don't think that exactly they will get it. Maximum similarities, the maximum similarities they will get it. Okay, based on that they can identify. Okay, even though their own children only, you don't think that 100% it will matches. The maximum similarities, okay, and dissimilarities. That's what I said. The DNA fingerprinting is comparison of the DNA from different sources to establish the identity. Okay, and see here the exactly solving the crimes with the help of DNA fingerprintings, how they will do the complete process, and finally it is useful. Okay, finally it is useful to identify the criminals which is involved in the different the criminal cases like murders, robbers, rapists, etc. Okay, so are you getting? So here the two individuals DNA is given, the DNA from individual A, DNA from individual B. Consider the DNA from individual A is a male parent, or the DNA from the individual B is a female parent. Then so here the chromosomes also they have mentioned clearly from the chromosome 7, chromosome 2nd, and chromosome 16, so that they can identify the similarities and dissimilarities, and based on the number of short tandem repeats. Okay, so and finally amplified repeats separated by the size on the gel given DNA fingerprints. So this is how it is useful during the solving crime with help of DNA fingerprinting technology. Okay, so let it be. So these are all with respect to the, so that means the DNA fingerprinting technology. So very important one, remember it is useful in the determination of genetic diversity as well as the evolutionary biology also. Okay, so that's why nowadays we are using this technique widely throughout the world, and it is also number of cases. Some people they will reject that, they will say this is not my child, and if they are having any the disputes with respect to the paternal issues or the maternal issues, then they will go to the the court. So in that court also they will suggest to conduct a DNA fingerprinting technology. Based on that they can solve all those problems. Okay, So let me to tell you briefly. So what are the things we were discussed today? We were discussed about the meaning of the DNA fingerprinting technology. So that is nothing but it is an analytical technique used in detecting the comparison, that is a comparing specific nucleotide sequences in DNA of individuals and thereby identifying the similarities and differences in such sequences. Majorly the steps involved in DNA fingerprinting technology, there are 6 steps. I said no, that one, first one the collection of the samples, the second one extraction of the DNA, the third step separation of DNA fragments, the fourth step extraction of the DNA fragments, fifth one the tagging radioactive DNA probes, and finally the sixth step autoradiography. Okay, so by using all these steps, so we will conduct a DNA fingerprinting profiling also, so that based on the applications we can utilize these techniques. Okay, so let it be. Thank you. Have a nice day.